Now I'm going to talk about chromatography. What is chromatography? Chromatography is a laboratory technique used to separate the components of a mixture that works by passing the mixture through a stationary phase, which is material, while another substance, which is in mobile phase, carries the mixture along. The mixture moves through filter paper and different parts travel at different speed. This method is low cost, uses very small amounts, and is very useful in science. It is also commonly used to separate amino acid, sugars, and pigments. Lastly, each substance travels a certain distance, which gives its RF value, a unique number for each substance. They also have different types of chromatography. Firstly is paper chromatography. Second is thin layer chromatography or TLC. Thirdly is column chromatography. Fourthly is gas chromatography, and lastly is liquid chromatography.
Today, I'm going to talk about flower chromatography. Flower chromatography is a simple method used to separate and identify the different pigments found in flower petals. Even though we usually only see one main color in a flower, it actually contains many hidden pigments. This technique helps us to see all those colors. So how does it work? We start by crushing flower petals and applying the color onto filter paper. Then, we place the edge of the paper into a solvent, usually water or alcohol. As the solvent moves up the paper, it carries the pigments with it. Because different pigments move at different speeds, they spread out and form a color patterns. This process is useful in science experiments, and it's also a fun way to explore the colors in nature. In short, flower chromatography helps us understand that what we see on the surface is not always the whole picture. To get the best results in flower chromatography, it's important to choose the right flowers. We can actually use almost any colorful flower, but flowers with deep or bright colors usually work best. These have more pigments, so the separation is clearer and more vibrant. Some great flower choices include hibiscus, marigold, rose, petunia, and so on. These flowers tend to produce strong, visible bands of colors when tested using chromatography. So, if you ever want to try this at home or in class, go for flowers with rich, bold colors. They will give you the most magical results.
Before we begin the experiment, I will explain the materials and apparatus needed for the procedure. First of all, flowers. Any type of flower can be used. It doesn't matter what kind or the color. Since flowers are solid materials, we need a solvent to extract their pigments. Acetone or methanol can be used during the grinding process to help to release the flower's pigments. For the solvent used to soak the chromatography paper, we can use water, chloroform, petroleum ether, or alcohol. Next, let's go over the apparatus required. A beaker or chromatography jar to hold the solvent. Chromatography paper, since we are performing paper chromatography. A ruler to measure the distance between the starting dot and the final position of the pigments. A plastic wrapper to cover the jar if the solvent evaporates easily, such as alcohol. A mortar and pestle to grind the flowers and extract their pigments. A stapler to help secure the paper in place if needed. A dropper to apply the flower extract onto the paper. And finally, a pencil to hang or suspend the paper, as pens should not be used as they may interfere with the results.
Next, I'm going to explain on how to do the experiment. First, take a strip of filter paper and draw a horizontal line about 2 cm from the bottom using a pencil. This is called the baseline. As a precautionary step, never use a pen as it might interfere with the result. Next, grind the flowers using a mortar and a pestle. Add a small amount of methanol or acetone to help dissolve the pigments. Stir and filter the mixture into a clean test tube. This colored solution contains the pigments that we want to analyze. Third, using a capillary tube or a dropper, place a drop of the filtrate onto the baseline of the filter paper. Let it dry, then repeat 2 until 3 times to make the spots more concentrated. Then, pour about 20 ml of the solvent mixture into a beaker or a chromatography jar. The solvent should be below the baseline to avoid direct contact with the spot. Hang the filter paper in the jar and cover it. Next, let the solvent rise up the paper. As it moves, it carries the pigments with it. Once the solvent front reaches 2/3 of the paper's length, remove the paper and then mark the solvent front immediately. Let the paper dry, then outline the colored spots using a pencil. Measure the distance each spot traveled from the baseline, as well as the distance to the solvent front. Then, repeat step 1 until 6 for the other flower sample. Lastly, calculate the RF value for each pigment using the formula distance traveled by solute divided by distance traveled by solvent.
Lastly, there are several ways on how flower chromatography works. Firstly is by pigment extraction. The flower's pigment dissolves in the solvent. Next is capillary action. The solvent moves up the paper by capillary action, carrying dissolved pigments. Then, a solvent like water or alcohol is added to the bottom of the paper, and it will move the pigment upward by capillary action. And lastly is separation by affinity. Pigments with higher solubility in the solvent move faster and farther. As the solvent moves up, it dissolves the pigments. The different pigments travel at different speed depending on solubility and absorption. Moreover, pigments with stronger attraction to the paper will move slower and less far.
Now, I explain some other uses of flower chromatography in real-life industry. First, forensic science. Chromatography is used to analyze samples from crime scenes to identify substances like drugs, explosives, or trace evidence. This helps investigators match evidence to the suspect and understand what happened at the scene. Next, the pharmaceutical industry. Chromatography ensures that purity and identity of drug compounds and helps detect impurity in medicine. This is very important to make sure medicines are safe and effective before they are used by patients. Third, food science. It helps identify and measure components in food and beverage, detect any harmful contaminants, and make sure the product meets quality standards. For example, it can be used to check if the color in food comes from natural source or artificial ones. Lastly, the perfume and cosmetic industry. This method is used to analyze the ingredient in fragrance, making sure product has the right scent and consistent quality. It also helps in creating new formula by studying different combinations of fragrance compounds. Flower chromatography is not just something we do in experiment. It actually plays an important role in many industries around us.
So, in conclusion, flower chromatography is a simple yet powerful technique used to separate and identify the pigments found in flower petals. By using suitable solvents and absorbent paper, we can observe the different pigments such as chlorophyll, carotenoid, and anthocyanin, each of which contributes to the flower's color. This experiment not only demonstrates the diversity of plant pigments, but also highlights how chromatography is used in scientific research to analyze complex mixtures. Through these activities, we gain a deeper understanding of plant biochemistry and the role of chromatography in analytical science.